Assalamualaikum and good day to everyone. So kita bertemu kembali for the third slot, revision and discussion for the answer screen knowledge booster. And today we are going to discuss chapter 5, which is the cellular respiration, and also chapter 6, photosynthesis. Okay, for your information, this two chapter is one of the major chapter in our PSPM. So, di mana untuk cellular respiration lebih kurang 15 markah. Okey, bukan sikit 15 markah. Dan untuk photosynthesis lebih kurang 9 markah. Okey, jadi bentuk soalan mungkin boleh pelbagai. Mungkin boleh essay, short essay, long essay ataupun structure. Okey, jadi kita tumpukan dulu answer scheme for the knowledge booster dan pada masa yang sama kita buat perbincangan sedikit fakta-fakta yang perlu anda tahu semasa menjawab soalan cellular respiration dan photosynthesis. Okey, kita teruskan. Alright, so ini adalah soalan kita. Okey. So, untuk soalan cellular respiration, so kita lihat soalan pertama dulu. Alright, in figure 1, it represent the main stage of aerobic respiration. Okay, so here dia dah bagi the keyword which is the aerobic respiration. Alright, so what you can understand by aerobic respiration? So, dia mesti ada presence of oxygen. Okay, presence of oxygen. Okay, so if you look at here, okay, so before we answer the question, so let's identify or analyze first the diagrams given. Okay, so the first one, okay, right in here is the glycolysis. So as you know, from glycolysis, from glucose, they will undergo the glycolysis process until they will produce the pyruvate. So this is the product from the pyruvate, which we will get ATP and also NADH. Okay, and then we continue from pyruvate to Krebs cycle. Okay, so don't forget from the pyruvate to Krebs cycle, and this one is the aerobic respiration. So the presence of oxygen so can continue to link reaction. So tulis siap-siap. Alright, so this is on the way to link reaction, and this one is the Krebs cycle. And from the Krebs cycle, as you know, they can produce NADH, ATP, FADH2. Okay, and actually NADH and FADH2 able to enter the oxidative phosphorylation. Okay, so here they only show the electron transport chain sahaja. Okay, so analyze dulu. So A, B, C. So what is the location? So as you know, the glycolysis will occur at cytoplasm ataupun cytosol. Alright, so our Krebs cycle, because they need the enzyme. Jadi, bila dia perlukan enzyme, so dia akan berlaku dalam matrix of mitochondria. Okay, kenapa kena sebut juga perkataan mitochondria? Because kita ada a few organelle yang contain with the matrix. So maybe matrix of chloroplast. Jadi better kita tulis nama penuh dia matrix of mitochondria. Okay, and for the electron transport chain. Okay, so kita tahu dia adalah a series of electron transport chain. So dia perlukan protein yang embedded. So this one will be the inner membrane of mitochondria. Okay, samalah. Mesti kena ada of mitochondria tu tadi. Alright, so done. Okay, so now let's have a look. What is the question asking about? Okay, state precisely where the reaction in the boxes. Okay, done. Kita dah jawab dah pun. Okay, so tengok ini kelebihan kalau kita analyze dulu kita punya diagram dan kita senang untuk terus jawab soalan. Okay, so dah ada A will be your cytoplasm, B will be your matrix of mitochondria, dan C will be inner membrane of mitochondria. Okay, yang ni kalau membrane sahaja akan jadi salah mesti specific inner membrane. Okay, what is X? Dekat sini kita tak jawab kan? Oh, sorry. Okay, so what is X di sini? Alright, as you know, dalam kita punya Krebs cycle, kita ada banyak proses dan antara proses yang boleh menghasilkan NADH is what? Yes, dia adalah proses oxidation and decarboxylation. Okay, so kita faham bila ada je perkataan decarboxylation, so apa yang mesti dikeluarkan di sini? Yes, dia mesti akan keluarkan CO2. So your answer here will be carbon dioxide. And alright, a total of 38 molecules of ATP are formed during the complete breakdown of one molecule. So this one is the considered keyword. One molecule of glucose, 38 ATP. Okay, so how many net? Nampak? Okay, so sekejap saya highlightkan yang ni. Okay, so net ATP. Dia nak yang net dalam 38 molecule from one glucose. Okay. So for each stages, so di mana yang ni adalah glycolysis, Krebs cycle and ETC ataupun oxidative phosphorylation tu tadi. Okay, dah ingat mesti net. Kenapa saya highlight pasal net ni banyak-banyak? Sebab kita faham dekat pyruvate you might get confused. Sebab dekat pyruvate kita ada dua stage, betul tak? The first stage is energy investment. Okay. And the second one is energy payoff. Okay. So dekat energy payoff, okay, kita sebenarnya all together dapat 4 ATP. And masa kita buat energy investment, kita sebenarnya guna atau hutang 2 ATP. Okay, so kita hutang 2 ATP. Alright. Okay, so here 4 ATP. Jadi bila soalan dah siap bagi tahu, dia nak net. So for your A should be berapa ATP? Yes, 2 ATP. Jangan 4, salah. Okay, alright. For your second one, Krebs cycle. Okay, so kita tahu dekat Krebs cycle sekali saja oxidative substrate level phosphorylation berlaku. Kan? Jadi dekat sini pun 2 ATP akan dihasilkan. Okay, dan yang ketiga, electron transport chain. Kita tahu the major ATP dihasilkan di sini. So kita tahu semuanya adalah 38. So 4 dah ada. Balance mana? Balance. So kita ada 34 ATP lagi di mana dihasilkan dalam ETC. Okay, sorry, oxidative phosphorylation. Boleh? Okay, kita teruskan. Energy is released during the electron transfer. Okay, so what is the general name? This one is a little bit your chemistry. Apa the chemistry terms you use when the energy is released? You are good. You are better than me, I think, because this one is the chemistry question. So the answer is exergonic. Okay, betul tak? Okay. Kalau endogonic means energy in. Yang ni kita release the energy. Okay. Alright. Sebenarnya soalan 2 and 3 ni sama, betul tak? Tak. Tengok ni. This one is the electron. Okay. So I highlight dulu kat sini. Okay. Right. So this one they nak what is the final electron acceptor, and this one they nak final proton acceptor. Jangan terconfuse. Okay. So final electron acceptor will be siapa? Our oxygen. Okay. So bila dia combine with the oxygen, so dia yang akan form apa? Water. Okay. So bila dia kata final proton, our proton kat sini sebenarnya dalam bentuk H plus, betul? Okay, so kita ada semuanya berapa? 2H plus lalunya kan? Jadi, siapa yang akan bergabung dengan 2H plus ni? Ya? Yeah? Yes, kita akan bergabung dengan oksigen. So, dia pun sama. Bila oksigen combine dengan kita punya 2H, baru kita boleh dapat water tu tadi, betul tak? Okay, so sebenarnya yang ni just to your information. This one not your answer. Your answer hanya oxygen. Same goes for the down one. This one your answer is only oxygen. So sebenarnya madam just tulis untuk menunjukkan yang oksigen tu at the end bila bergabung dengan hydrogen, so they will be formed as water. Okay. So now let's go to question number 2. Okay. Alright. So figure 2 show the stages of cellular respiration occur in cytosol of the cell. Alright. So what is the keyword here? Kita nak tahu ini adalah proses apa. So look at here. Given here is the glucose. Okay. Dan kita nampak dia berlaku dalam cytosol. Alright. Dan apakah produk dia? Okay. So dia tak tulis nama produk. Tapi dia bagi apakah komponen yang kita dapat. And then baru dia masuk kepada mitochondria. So it's easily understood here. This one is the 3 carbon of pyruvate kita. Okay. Berapa dekat sini ada 2 molekul. Okay, so jangan lupa dekat sini. Okay, our DHAP will be isomerized. Okay, and form as a glyceraldehyde 3-phosphate. Okay, so now name the process in figure 2. So obviously this one is the glycolysis. Okay. Name the enzyme that catalyze this reaction. So bila dia kata catalyze, okay, as soon as the glucose molecule enter the cytosol, a phosphate group is attached to the molecule. So maksudnya dekat mana? Okay, it's this one. So dekat sini nampak? Alright, so they are using the ATP. So kita ada 2 ATP semuanya. Okay, tapi dekat sini dia kata as soon as glucose molecule enter. Jadi dia bukan nak yang kedua, dia nak yang pertama. Because as soon as glucose enter. So this one. What is the first enzyme? Yes, kita ada hexokinase. Okay, kalau yang second one, phosphofructokinase. Tapi dekat sini kita tak boleh jawab because dia nak immediately after glucose enter. Okay. State the class of this enzyme. Okay, look at the characteristic given here. Okay, so dia sebut apa? A phosphate group is attached. Maksudnya, nampak dia pindahkan. Okay, so bila dia pindahkan, so dalam kita punya biocatalysis, bila dia able to transfer the molecule ataupun transfer the phosphate group in this example, so kita sebut dia sebagai transferases. Ini nama enzim dia. Okay, transferases. Okay, sebab dia boleh transfer the phosphate group tu tadi. Okay, now molecule A, B, C and D. So kita lihat one by one. What is molecule A? Okay, so tadi madam dah sebut kan? Short form is a DHAP. Another one is glyceraldehyde 3-phosphate. But dalam exam tolong jangan buat short form. So namanya dihydroxyacetone phosphate. Okay. Kejap oxy. 1O. Okay. Okay, B. Mana B kita? Alright. So daripada glyceraldehyde 3-phosphate, so kita nampak sekarang dia ada 2 phosphate. So namanya akan jadi 1,3 sebab phosphate berada dekat carbon number 1 and number 3. And sebab dia ada 2, so this phosphoglycerate. Hati-hati dengan spelling madam pun kena hati-hati ni. Bisphosphoglycerate. Okay, C. Alright. So nampak bila water is removed sebelum dia nak jadi pyruvate, so this one will be phosphoenol pyruvate. Kena tulis nama penuh Phosphoenol pyruvate. Okay, and your D will be the last product which is a pyruvate. Okay. Okay. Boleh. Senang Okay. What is the types of phosphorylation shown in step 5? Kita tengok. Which is the step 5. Okay. 4 and 5. Kita nampak dia boleh hasilkan ATP. Okay. Kalau kita recall balik, kita hanya ada 2 phosphorylation, iaitu substrate level phosphorylation. Dan satu lagi kita ada apa? Oxidative phosphorylation. Dua proses ni sahaja. Okay, so obviously kita tahu substrate level phosphorylation ni boleh berlaku dalam glycolysis dan juga Krebs cycle. Okay, sebab dia ni yang boleh menghasilkan, kita tolak ni sikit. Tolak kat mana? Tolak bawah sikit. Okay, alright. So kalau oxidative phosphorylation dia ni yang berlaku di mana? Yes, ETC plus chemi osmosis kita tu kan. Right? So kita boleh ingat terus. So kalau oxidative phosphorylation dia hanya berlaku di sini. Kalau glycolysis and Krebs cycle, substrate level phosphorylation. So this one, the answer will be the substrate level phosphorylation. So kita copy dan terus jawab di sini. Tapi belum habis. Titik. Sebab dia nak explain the reaction. So if you look at the chapter stage 5, kita kena explain lah what is happening until the ATP can be produced here. Okay, so you can start your word by, okay, so dekat sini, apa yang jadi? So the high energy phosphate in mana? Kita bertukar dari molekul mana? Tengok balik Alright. So from 1,3-bisphoglycerate. So in 1,3-bisphoglycerate, okay, is transferred to ADP to form ATP. Okay, baru 1 mark. Okay, the next mark, markah yang ketiga. Right? So, apa jadi pada 1,3-bisphoglycerate kita? So, 1,3-bisphoglycerate converted into, jadi apa? Tinggal satu dah kena buang. So, 3-phosphoglycerate. 3 markah So, 1 markah for the substrate level phosphorylation sebab dia tanya what type. Okay, then markah kedua dan ketiga you are explaining. Okay, so kalau kita tengok daripada gambar ni, alright, so 1 phosphate daripada 1,3-bisphoglycerate dia akan transfer the phosphate pergi kepada ADP. That's why dekat sini kita boleh buat contohnya tambah phosphate inorganik kan, PI. Jadi bila yang ni tambah PI kat sini sebab datangnya daripada 1,3-bisphoglycerate, so that's why dia boleh jadi ATP, triphosphate 3. So daripada 1,3-bisphoglycerate now dia akan jadi 3-phosphoglycerate. Itu dia. Okay, so selalunya kalau soalan mirip macam ni memang dia suka tanyalah. Okay, so this is the method or the way you to answer. Okay, so let's go for the third question and last question for the cellular respiration. Okay, once again, aerobic respiration is occur. So now let's identify. So glucose, what is stage 1? So you know stage 1 will be the glycolysis. Okay. Alright, then what will be the compound? Means what is the product from the glycolysis? So it's a pyruvate. Okay, then what is the stage 2? Kita tahu from the pyruvate, sebab dia kata presence of oxygen kat sini, so they will undergo the aerobic respiration. So this one will be the link reaction sebab dia on the way from cytosol enter the mitochondria here. Okay, so apakah hasil link reaction kita? Acetyl CoA. Okay, ingat kalau nak jawab CoA short form, you must write C capital letter, O small letter, A capital letter. Okay, ataupun katalah dalam exam alamak terlupa, so you can write in full sentence which is a acetyl coenzyme A huruf besar. Samalah sebenarnya. Boleh pilih mana-mana satu. Okay. Then this one from compound A acetyl-CoA. So dia masuk kepada stage 3. So this one will be the Krebs cycle lah. Okay, then. Okay, so name the stage 3. Kita dah jawab dah. Krebs pula. Macam ingat kebab pula Okay, Krebs cycle. So this one will be the identify compound A and B. Senang. Kita dah tulis siap. So this one will be the pyruvate. Okay. Okay. And B will be your senang. So bestnya kalau exam boleh copy and paste macam ni kan. Okay. Alright. So pyruvate and acetyl-CoA. Alright. Once again. Specifically, which part of a cell does stage 3 take place? Okay, tengok balik. Where is your stage 3? Krebs cycle. Okay, tadi saya dah bagi tahu. Krebs cycle, we are using the enzyme. So, they need the fluid. So, this is the matrix. What happened to a compound A in the presence of oxygen? Okay, maksudnya kat sini kan, compound A, presence of oxygen. So, kita sebenarnya jawapan dah diberi kat sini. Perasan tak? Pyruvate will continue buat apa? Converted into acetyl-CoA. Lepas tu acetyl-CoA pula akan continue buat Krebs cycle. Sebenarnya jawapan memang dah ada dalam kita punya soalan diagram ni. Okay, so kita salin je balik. So, what happened? The pyruvate converted into acetyl CoA. Okay. Converted into acetyl-CoA. Then what the acetyl-CoA will do? Continue the Krebs cycle. Ambil sebiji je daripada sini. Nampak? Itulah pentingnya bila kita conteng soalan sebelum kita jawab. Semua ada di situ. Okay, see. Name the 2 electron carrier used in stage 3. Okay, dekat stage 3 maksudnya electron carrier yang paling glamour kita ada NADH. Okay. Dan kalau dekat stage ataupun dekat Krebs cycle kita dapat satu lagi FADH2. Okay, ingat 2 ni kena kecilkan kecilkan size 2 ni. Oh, dia kecilkan semua pula. Okay, so kecilkan 2 ni Okay, in stage 3 involve the conversion of isocitrate to alpha ketaglutarate. Okay, if you look at here, dia dah siap-siap bagi this one, here saja dia nak. Okay, ni saja. So, dekat proses apa yang isocitrate will be changed into alpha ketaglutarate? Yes, it's a oxidation and decarboxylation. Okay, dah ada jawapan sini. Okay. Okay. Now, briefly explain the chemical reaction that occur during this conversion. Maksudnya kita nak ceritalah. Kalau dekat sini kita dah siap tulis dah, oxidation. Now, kita just bagi tahulah siapa yang sebenarnya telah dioxidize. So, kita tahu isocitrate is oxidized. Okay. Jadi, kalau seorang dah oxidize, siapa yang di-reduce? Okay, so NAD plus is reduced to NADH plus H plus. Okay, alright, satu markah. Jadi, apa saja jadi? Okay, dah oxidation kita dah cerita. Sekarang jangan lupa decarboxylation. So, 1 carbon is removed as carbon dioxide. Okay. In proses apa? Decarboxylation. Okay. So bila soalan tanya macam ni, just kita tengok je. Isocitrate kepada alpha ketoglutarate. Apakah proses yang terlibat? So sebenarnya itulah chemical reaction yang sebenarnya berlaku dalam soalan ni. Okay. Settle untuk cellular. Now, the chapter 6. Okay. So just now I told you chapter 6 about 9 questions will come up in your final PSPM. Okay. So let's discuss for the chapter 6. Okay. For chapter 6, this one is the light dependent stage of the photosynthesis. Okay, can you recall back what is the light dependent? Yes, those yang involve the PS1, PS2. Okay. All right. So then when we are using the sunlight, so the sunlight is changed into the energy photon. Okay, so they will activate the electron. Okay, then electron become excited, catch by the primary electron acceptor, and then they go through a series of electron transport chain. But here, it's not required oxidize, reduce semua tidak perlu. But we understand that when ETC is occur, ATP will be produced here. Okay, then and once again, the second time electron will become excited and catch by the primary electron acceptor. And now they will be using the one enzyme to produce a NADPH. So all the product come from the light dependent will be used in light independent. Okay, so dah cerita banyak dah. Alright, so what is P, Q, R, and S? So if you look at here, P, Q, R, and S. So this one all is the protein in the ETC. So the first one, kalau you still remember, short form dia is P, Q. Oh, sorry, P besar, Q. This one, ni, P, C. Yang ni, F, D. Senang kalau kita ingat dia punya nama kecil, nama pendek dia. So this one is plasto, Q is quinone. So what is your R? Plastocyanin. Sebab C kan? So plastocyanin. Okay. This one, cytochrome complex. And the last one is the FD, perodoxin. Where does light dependent stage of photosynthesis occur? So kita tahu which structure in our chloroplast yang consider as a capture of the sunlight. Yes, thylakoid membrane. Okay, so thylakoid membrane. Name the reaction that involve in splitting of sugar. So kalau nampak kat sini, perasan tak? The water is split into oxygen and hydrogen. So this process will release the electron. So electron ni yang kita recycle kat sini kan? Yang kita gunakan untuk jam. Alright, so it's known as photolysis of water. Jangan photolysis saja, photolysis of water. Photo represent the presence of light, so they undergo the hydrolysis dan dia pemecahkan water. State 2 product of light dependent reaction that is needed to keep the Kelvin cycle running. So sebenarnya they are asking what is the product from our light dependent reaction is shown here. So kita dapat ATP dan NADPH. Okay, so catat je balik. So we get ATP. And NAD, jangan lupa ya, this one is a photo. So pH. Okay, so menandakan dia adalah untuk foto. So plus H plus. State the wavelength. So dia nak wavelength ya. As you know, PS1 is also known as what? P700. Yes, correct. Tapi soalan nak wavelength. So berapakah kapasiti wavelength dia? So 700 and you must put the unit dia, nanometer. Alright. So this one also as you know is a P680. Tapi sebab dia nak the wavelength, so 680 nanometer. Okay. So now let's go to question number 2. Okay. So shows the light independent reaction in photosynthesis. Okay. So in which part of chloroplast this occurs? So kita tahu this one is what? It's a Kelvin cycle. Okay. Dan kita tahu Kelvin cycle dia akan menggunakan Rubisco. Okay, which is an enzyme. Jadi kalau dia menggunakan enzyme, so dia mesti dalam fluid dia. So fluid yang kita jumpa dalam chloroplast namanya stroma. Okay, what is phase 1, 2 and 3? So phase 1, carbon fixation. Phase 2, reductions. Kita reducekan kat sini kan. And phase 3, regeneration of RUBP. So carbon fixation. Okay. So this one, reduction and regeneration of RUBP. Okay, kita boleh guna RUBP tapi dengan syarat mesti tulis R besar, U kecil, BP besar. Okay. Ataupun ribulose bisphosphate. Okay, boleh. Yang ni short form boleh ya? Boleh terima. Name the molecule P and its function. Mana dia P? Ni dia. Okay. So maksudnya during reduction, selain daripada ATP, there's another one yang we use here. So this one is the product come from the light dependent. So what is it? So it's a NADPH. Okay, satu markah di situ. Now, apa function dia? So dia sebenarnya sebab dia adalah nama pun proses reduction kan. So dia reduce. Reduce apa? 1,3 bisphosphoglycerate. 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 Okay. To become glyceraldehyde triphosphate. Dia punya molekul tu be careful ya sebab kadang-kadang kita nampak dia macam kejap dekat crab macam ni juga, dekat Kelvin pun macam ni. Tapi sebenarnya kalau dekat foto kan nampak dia ada bisphosphoglycerate. Okay, dia tambah lagi sikit. Alright, so 2 markah di situ. Okay. Identify the R. Mana pula R? Okay, kita tahu untuk Kelvin cycle, so inilah gunanya CO2 kita, betul? So, kita akan guna CO2. Okay. And what is the enzyme that able to involve in forming the 3-phosphoglycerate? Okay, so siapa sebenarnya yang boleh enzyme catalyze the formation? So, kita akan guna Rubisco. Okay. Yang ni. Ataupun RUBP carboxylase. Okay. Nama lain dia RUBP carboxylase. Pun boleh. Okay. Tapi kalau dah ada bagi short form ni boleh diterima. So guna je lah yang ni. What is the relationship between the light dependent reaction and light independent? So kita faham ATP, tadi kita guna dekat sini NADPH, semuanya datang daripada light dependent. Dan hasil daripada light independent which is ADP, nanti kita dah tukar. Dan dekat sini NADP+, semuanya kita akan pulangkan balik kepada light dependent. So, macam mana nak buat ayat? So, ATP and NADPH. Okay. Which is come from light dependent, okay, will be used in light independent. Okay, alright. Kita 2 markah. So, yang hasil daripada light independent kita akan dapat ADP and NADP+ will be returned back. light dependent. Supaya dia boleh gunakan semula. So itu dia macam recycle lah. Okay. You give, I give. Macam itulah konsep dia. Okay. Soalan nombor 3. Okay. And last one. Alright. Alright. So before we start answer, CAM. So little bit idea about the CAM. Anything that you can recall. Okay. CAM. Sebut je kita dah terus boleh ingat. Cactus. Cactus. Dia will open the stomata during night sahaja. So enough this keyword. Awak dah boleh faham You dah boleh bezakan dah. Okay. So night and day. Okay. So kita nampak during night baru CO2 akan masuk. Sebab during day dia memang tutup stomata. Okay. Then daripada apa? Okay. So from molecule phosphoenol pyruvate. Okay. Phosphoenol pyruvate. So dia akan menjadi apa? Malik. Okay. Dan kita tahu dekat wakul ni sebenarnya malik ni malik ataupun boleh juga jadi malik acid sama malik acid. Okay. So dia akan simpan semalaman dan during day dia akan convert back malik. Okay. So baru dia akan masuk. Nampak kalau pusing macam ni. So this one will be the Kelvin cycle. Okay. Dan kita tahu apa yang boleh dikeluarkan through the Kelvin cycle. Okay. So inilah kita punya oksigen akan dikeluarkan di sini. Dah. Kita dah tafsir habis. So sekarang kita boleh jawab. Apa dia P? Okay. Dah jawab dah pun. Phosphoenol pyruvate. Right. Transfer here. Okay. What is Q? Malik. Silap. Okay. So Malik. Malik. Kat bawah. Okay. Dan bawah R is what? Sorry. P. Oh, Madam Ternate. Tak apa. Saya turunkan saja. Wah, senang soalan macam ni kan? Okay. So dekat sini sebenarnya adalah oksigen. Okay. Oksigen. So Q kita adalah phosphoenol pyruvate dan malik. Okay. State the advantage of CAM. Okay, kita tengok balik. Kita tahu kita punya cactus selalunya durung kat gurun Sahara, duduk dekat yang very dry and hot. So sebenarnya apa kelebihan CAM? So actually they can allow the plant to grow in hot and dry condition. Okay, sebab kalau kita tengok pokok kita sekarang kan, kalau musim panas dah mula kering, mula lembek dah. Okay, tapi kalau CAM plant, they can survive. One example, okay, so yang commonly ataupun yang paling glamour, cactus ataupun pineapple. Okay, alright, so name the process X. Okay, kita dah siap dah tafsir dia, which is the Kelvin cycle. Okay, tulis siap. Right. Then the next question, what will happen if there is a high concentration of oxygen? Okay, kita faham. Bila dekat Kelvin cycle, our Rubisco will tend to bind any of the oxygen or CO2 yang present. So kalau dia bind dengan oxygen, so what happen? The photorespiration will occur. So kita tahu bila photorespiration occur, apa jadi? Dia dah tak boleh menghasilkan the glucose lah. Dia dah macam fail the process of the Kelvin cycle. Okay, name the pathway of carbon fixation in C4 plant. Okay, so nama dia apa? H slack pathway. And for your information, nampak H dengan S pastikan huruf besar. H slack pathway. Okay, compare. Alright, ingat bila soalan compare, jangan pula awak buat perbezaan je. Dia mesti ada similarities and differences. Always remember this one, please. Soalan dalam bio dia memang glamour, similarities and differences. So kalau dia bagi 1 markah, so boleh bagi 1 similarity, 1 differences. Kalau dia bagi 3 markah, satu similarity, dua differences. So selalunya differences ni kita lebihkan sikit. Please kalau soalan compare must consist of similarity and differences. So first, what is the similarity? Okay, so yang kita boleh think both undergo a Kelvin cycle. Okay. Apa lagi? Both 2 times carbon fixation kan. So both pathway undergo twice carbon fixation. Ini saja saya bagi pilihan dua. Okay, now one more differences. So the CAM carry out carbon fixation in different, perasan tak? Dia akan buat dekat different apa? Night and day. So this one kita sebut different time. While in C for plant, carbon fixation in 2 different types of cell. Mesophyll cell dan bundle sheet cell. Nak cerita pun boleh lah. Okay. Mesophyll cell and bundle sheet cell. Okay. Alamak. Turun bawah. Sada. Done. Boleh? Okay. So that's all for now. Okay. So we already discussed 2 chapter. This is chapter 5. Okay. Cellular respiration. Ini dia. Alright. Cellular respiration. Okay, so di mana cellular respiration, we expect about 15 markah dalam PSPM. And second one, we are discussing about photosynthesis. So kita expect dalam 9 markah. So paling banyak sebenarnya cellular respiration dan photosynthesis. Okay, in next discussion, kita akan fokus gases exchange and transport. Okay, see you. Bye.